Welcome to AP Biology. Today we're talking about cellular respiration and we are at the Krebs cycle step or the citric acid step. So previously we've talked about how glucose can get cut in half in glycolysis to make pyruvate, two pyruvates, which are 3 carbons big. And then this is the pre-Krebs step, where glucose is, well, it's not glucose, it's a half of glucose. Glucose is oxidized some more, making some more NADH, and you break off one of these carbons and you make carbon dioxide, and then you use this CoA thing, and you make acetyl CoA. Acetyl CoA is what enters the citric acid cycle, and it's called a cycle because it continuously regenerates the molecule that you need to get started. So what happens to the acetyl CoA? Well, there's 2 carbons going in, and you end up making carbon dioxide. So there are some hydrogens on here. What happens to those? Well, the hydrogens and the electrons get ripped off or oxidized, and you produce more NADH. You also rip off some electrons and hydrogen ion protons to make FADH2, and you make some ATP. So that's sort of the summary, but what I want to do is show you all the little steps and then come back to the summary in a little bit. But the big picture is that you started with glycolysis and then pre-Krebs by oxidizing glucose. When you oxidize it, you pull off the electrons and make NADH. This Krebs cycle right here, the citric acid cycle, it completes the breakdown of that half glucose, of that pyruvate, into carbon dioxide. You've broken all the carbon-carbon bonds, so all you have are individual little glucoses. You've broken all the carbon-carbon bonds in glucose, so all you have are these individual little carbon dioxides at the end, and you've ripped off all those hydrogens. So remember, glucose is C6H12O6, and so you've made a whole bunch of carbon dioxides by the end of this, and you've ripped off those hydrogens, and those hydrogens have a proton and electron, and those have gone on to NAD plus to make NADH. So the Krebs cycle makes a whole bunch of those. 3 NADHs, 1 FADH2, and then a little bit of ATP by substrate level phosphorylation, just like I showed you before with an enzyme. All right, so where does the Krebs cycle take place? It takes place in the matrix of the mitochondria. So if you imagine a bacterium early on that was really good at cellular respiration, the enzymes to do all this stuff would be in here, right? The Krebs enzymes. And then remember that bacterium that was really good at cellular respiration was eaten up by a pre-eukaryote, right? An early eukaryote, till eventually it looked like this. And so notice there's a double membrane, but you would expect that all the Krebs enzymes would still be inside that, right? So that's sort of like the cytoplasm of the early bacterium. It's called the matrix of the mitochondria now. This is what's true for eukaryotes. So for eukaryotes, the Krebs cycle takes place in the matrix of the mitochondria, but you know that prokaryotes don't have a mitochondrion, so it just actually takes place in their cytoplasm. Here are all the steps. I think once upon a time, way back in 1980 something, or maybe early 90 something, I had to memorize all these. I have long since forgotten them, and you do not need to memorize any of them. But I want you to see that there's a whole bunch of structures and there's a whole bunch of chemical reactions, and each one requires an enzyme. You don't need to know the enzymes, but here's acetyl CoA. It's that 2-carbon compound. Remember, you've got glucose, which has 6 carbons, broken in half to pyruvate, which has 3 carbons, like 1, 2, 3. It's broken to acetyl CoA, you release a carbon dioxide, you've only got 2 carbons. Right here, you've ripped off some electrons, so you make NADH. Anyway, acetyl CoA combines with oxaloacetate. There's an enzyme that does this, Citrate Synthase. You don't need to know that. You make citrate, thus the citric acid cycle, right? Coolio, right? If you have this thing that has 1, 2, 3, 4 carbons, and you have this that has 2 carbons, you put those together, 4, 5, 6, this thing, 1, 2, 3, 4, 5, 6, that should also have 6 carbons. The grayish, blackish ones are the carbons. Take out some water, you get cis-aconitate, I think. Here's another enzyme, aconitase, makes isocitrate. You rip off some electrons, you're oxidizing the isocitrate, you make oxalosuccinate, and then you need the enzyme isocitrate dehydrogenase. Please don't memorize any of those. Ooh, look, you're ripping off some carbon dioxide. Anyway, each one of these steps requires an enzyme, and sometimes you're breaking off some carbon dioxide, which means if you start with glucose with 6 carbons, and then you go down to pyruvate with 3, because there's 2 of them, right? 2 pyruvates. And then you rip off 2 carbon dioxides here. You're ripping off another one here. But because you have 2 pyruvates, you're going through the whole thing twice. So really there's like 2 more there. Let's see, more carbon dioxide is removed. Right here. You're also ripping off some electrons to make NADH, right here. And you make FADH2 right here, and some more NADH right here. You do not need to memorize any steps of this, but just notice that there are a lot of steps, that each one will require an enzyme, and that at the end you end up making this thing that you needed in the beginning, right? You need oxaloacetate. That'll go together with acetyl Coa. You'll have all of these reactions take place until finally you have this same thing all over again. Meanwhile, what's the result? Well, if you had glucose with 6 carbons, let's see, let's make a glucose with 6 carbons. Let's see, this way. Okay, so here's my glucose with 6 carbons. So we did glycolysis and made this, right? Then pre-Krebs, you've got pyruvate here. Pre-Krebs will break off a carbon dioxide, and now you're left with acetyl CoA, and then it starts to go through. Remember, you're going to have these 2 both going through. So C, right? Where's my pen? See right here, you're breaking off a CO2. You're actually doing that to both of these. So that's 2 more carbon dioxides that have gone away. And then, but these are kind of stuck on another molecule now. But anyway, then you're breaking off 2 more carbon dioxides because the pyruvate goes through. There's 2 pyruvates to go through. So there's the rest of it. So what you end up doing, I really wish I could juggle, but what you end up doing is making 6 carbon dioxides by the time you're done with all this, which makes sense because you've broken glucose down completely. So glucose, remember, has a bunch of hydrogens on it. And those hydrogens have protons and electrons, right? And so you've broken that all the way down to 6 carbon dioxides. But what happened to all the hydrogens? Well, the electrons and the protons went on to NAD plus to make NADH, and also to make FADH2 from FAD plus. What are they going to do? Oh my God, I can't even wait to tell you. We are almost there. What is going to happen to the delivery molecules? So exciting. Notice right here, we've made a little bit more GTP, which gets converted to ATP. So that's by substrate level phosphorylation, because here's an enzyme that takes this and pulls the phosphate off, and you end up making ATP. So each step of the citric acid cycle is catalyzed by a specific enzyme. The acetyl group of acetyl CoA joins the cycle by combining with oxaloacetate, forming citrate. You don't need to know that. The next 7 steps decompose the citrate back to oxaloacetate, making the process a cycle. You should know that it's a cycle because you remake that same thing. The NADH and the FADH2 produced by the cycle relay electrons extracted from the food to the electron transport chain. Okay, that's the end of the story. It goes to the electron transport chain. I haven't taught you that yet, but what you should know is that these delivery molecules are taking electrons from food. So you start with food like sugar, and then the electrons from the food are going to be carried by a delivery molecule called NADH, or another one, FADH2. And then the food, the waste product, is going to be carbon dioxide. You're also going to make a little bit of ATP, so that's nice. But what happens to the NADH and the FADH2 is the subject of our next video. I know you can hardly wait, right? All right, so this is maybe a simplified version where you're not looking at each of the structures, but you can see how many NADHs you're making, right? You've got 3 NADHs made, but remember, acetyl CoA, there are 2 of them, right? Because glucose has 6 carbons, it's broken in half, so you'll end up with 2 pyruvates, 2 acetyl-CoAs. So really for each one glucose, you're making twice as many NADHs. You're making 6 NADHs. And then you're making a little ATP, 2 of them, and then 2 FADHs. I'm multiplying this 1 by 2, right? Because I have to do the whole cycle twice. And then here are the 2 carbon dioxides. If you multiply that, because pyruvate goes through twice, there's 4 carbon dioxides made, which makes sense because if you start with glucose, and you break it down to a half of that, right? Pyruvate just has 3 carbons. And then you break off 1 CO2 and you're left with this thing, Acetyl Coa. You've already broken off 2 CO2s because there's 2 pyruvates, right? And then you have 2 of these. They go through this whole cycle twice, which means you're getting 1, 2 times 2 is 4. So that's going to be, instead of 2 carbon dioxides, now we've got 4 carbon dioxides, altogether 6. Whoops. So that equals the 6 that we started with from glucose. So these are the delivery molecules. That's why I put the pictures in there. So notice that the delivery molecules have picked up some electrons and hydrogen ions. What's going to happen to them? What will they do with their electrons? You'll have to wait for the next video for that. It's exciting stuff. All right, summary. The citric acid cycle requires oxygen. You didn't see oxygen anywhere in here, but if you don't have oxygen, you can't do the next step, and then the whole thing would just stop. So even though it doesn't look like it requires oxygen, it really does, because these middlemen here, these delivery molecules, these delivery molecules need to give up their electrons at the next step, which requires oxygen. So you wouldn't do the Krebs cycle without oxygen. This is a cyclical series of reactions that give off carbon dioxide and produce 1 ATP per cycle, but you do 2 cycles for each glucose. Produces 2 ATP per glucose because you have glucose split in half, so you've got 2 pyruvates going through the whole thing. So here's your total net yield. You've got 2 ATP from the Krebs cycle, from 2 turns of the Krebs cycle. Remember, I think this is where a lot of kids are like, you've lost me. I don't know what you're talking about. Here's glucose, right? Glycolysis split glucose in half. And so now I've got 2 pyruvates. That's acetyl CoA. You've got 2 of them, so you have to go through the Krebs cycle twice, right? So for 2 turns of the Krebs cycle, you get 2 ATPs. Notice they each have, I'm trying to show you 3 phosphate groups here. Anyway, you get 6 NADHs, so it's kind of a lot when you line them all up. You get 2 FADH2s, and you get 4 carbon dioxides. You had already released 2 carbon dioxides earlier. Earlier, you had also made some NADH also and some ATP, remember? Like in glycolysis, you make a couple ATP and you make a couple NADHs, and then the pre-Krebs cycle. Okay, so evolution of the Krebs cycle. This happened later than glycolysis because it requires oxygen. So we needed an environment that had oxygen before this evolved. So does this make sense? Well, bacteria evolved 3.8 billion years ago or so, and that's when glycolysis evolved. But then free oxygen was only present about 1 billion years later because of photosynthesis. And so this glycolysis, this Krebs cycle thing requires oxygen eventually. And so it had to evolve after we had oxygen present in the environment. Okay, ATP accounting so far. Glycolysis made 2, Krebs has made 2. But life takes a lot of energy, and 4 ATP is not very much. So you need a lot of energy, and there's some better way. A working muscle requires 10 million ATPs per second. There is a process that makes a lot more ATP, and we're almost there. It's called aerobic cellular respiration, which we started, right? Krebs is part of that. So the better way is aerobic cellular respiration. You just wait for those delivery molecules. All right, and that is it for this video.